So our next session is around genetics and breeding, and we've heard a lot of our speakers stress the importance of developing the genetics in order to get our industry up and going. So we're going to have Jo Kerslake begin this conversation. Jo is a partner at Abacus Bio down in Dunedin, and she thrives on the challenge of improving the efficiency and profitability of agri-systems. She's got experience in both the sheep farming and dairy farming sectors, and she has a PhD from Massey University in Animal Sciences. Thanks, Jo.
All right, good afternoon. So for you astute ones in the audience, you'll realise my title of my talk has changed slightly. On Friday I had a look at the program and had a slight freak out that maybe we're all going to talk about the same thing. So today I thought I'd actually just change tack slightly and try to attempt to set the scene, I guess, to talk about what value genetic improvement adds, what the key considerations are in terms of a genetic improvement program, and I'm going to finish up with just a couple of key questions, I think, around sheep milk genetics and what it might take to get an improvement program for the industry. Right, the power of genetics, no words needed really. Genetics is an incredibly powerful tool, and we only have to really look at the poultry industry to see how targeted selection can really transform an animal in terms of getting to what market requirements need and in order of creating profit for the farmer. As Lucy mentioned, I work with Abacus Bio, and where practical application of genetics is really our core business. We have a number of quantitative geneticists, software developers and farm system consultants, and generally we all work together in order to help farmer and industry and improve profitability of businesses in the primary industry. So genetic improvement is simple really. The key principles really are around making sure you record the performance of animals, using high-performing animals as the parents of the next generation. The effectiveness of the programs is dependent on how heritable the traits you're looking at are, so what's the likelihood of receiving them from your parents, and how much variation exists within that trait. So if there's a lot of variation in the trait that you're looking at, then you're likely to make some reasonably fast gains. If there is a little variation in that trait, then your gains are likely to be a lot slower. So three core components that I think are required for a genetic improvement program really, there has to be a value proposition for the farmer. Selecting traits that are profitable in their farm system and for where they're heading in terms of market is crucial, and it's a non-negotiable. The other one is that the genetic program has to be simple to implement on farm, so traits have to be easily recorded. That data has to be able to be easily stored and accessible to geneticists who can run evaluations to determine who is the best of the best. And also, and the outputs that come out of those evaluations really do need to be easily interpretable so that they can be used on farm. The third one is a well-designed breeding program. So this is around structured matings and to make sure that the matings are structured in a way that you're actually accurately identifying the best individuals. And then the second pass is how we use those best genetics and how we can cost effectively distribute them across industry. So in terms of value proposition, I guess the breeding goals and breeding objectives are really a key place to start. So in terms of a breeding goal, the main things that you really want to ask yourself what is the particular task that I want to achieve, and what is the ideal animal to complete that task. The next two questions really comes about knowing your business. Where does my profit come from, and what are my biggest costs? And if you have a good handle on that, then it can help you develop an effective breeding objective where you're looking at the traits that you desire and understanding what traits are profitable earners and which are not. And then you can have a look at how those traits interact together in order to, how they interact and have an effect on overall farm profit. Genetic improvement programs really have yielded outstanding results in the New Zealand primary industry, and one example of that is the New Zealand dairy cow industry. We've looked at that and we've estimated that genetic gain's worth about $3 million per year. So in order just to, I guess, help you understand how genetics adds value, I'm just going to step through some examples of how we've got the $300 million figure. So genetics adds value because it's permanent. So if you have 1 year of genetic improvement, which you can see on the left-hand side graph, and then you do nothing else for the next 20 years, you will still have that gain during that time. And in addition to it being permanent, it's also cumulative. So if you do genetic improvement over the next 10 years and then nothing for the remaining 10 years, then you'll end up having a number of multiple benefits. So if we have a look at that graph, and if we think that one blue square is a gain, then you can see for the 10 years with a step up and then 10 years of no more improvement, that there are 155 gains during that time. But there's always a but. So while genetic improvement has multiple gains, it is a long-term gain. So the best way to think about this graph, I guess, is that what $1 is worth today is not the same as what it's worth in 20 years. So it's important that we take that time frame into account. And so in this situation, we can add a kind of discount of 7%, which brings our 155 gains down to 75. Gains also have to be disseminated. So if we have a look here, which is a gene flow diagram of the dairy cow, you'll see on the blue bars, the dairy cow herself, and her genes do not go into her calf until when she calves as a 2-year-old. Those genes will then start to be disseminated, but it'll be another 4 years before her calf calves and her genes start to disseminate through the population. So then, once we've got an understanding of the amount of gains, we can have a look at what each gain is potentially worth. And in the New Zealand dairy industry, it's currently worth around $10 per cow per year. So if we just step through and bring all of that together, how genetics adds value to an industry is that it's permanent and it's cumulative. It disseminates throughout the population. For every genetic gain, there is an improvement in profit. And if we multiply that over 740,000 heifers per annum, then that will come to $3 billion, which is roughly $300 million per year of genetic gain in this example. So the second core component is really simplicity of implementing it on farm. So the data required for the breeding objectives really need to be easy to record. So a parentage is one of those, and there are a lot of technologies out there now that can make it a lot easier to record parentage. And it really pays to look at your selection criteria, so which traits you're going to measure. And if we look at milk yield, if you want to improve milk yield, the selection criteria doesn't necessarily have to be daily milk yield. It can be other things like doing 7 to 8 herd test records, which can predict the daily milk gain. So how you base your selection criteria will be dependent on your system and what technologies you have in terms of what you'll be measuring to predict for that trait. It's really important that data can be easily stored and then taken out as well, so that geneticists can do their evaluations. There are some industry options out there. So at the moment we have SIL, which is a sheep and beef improvement database. So I don't think currently it has anything, doesn't currently record any milk, but I know that Suzanne Rowe is working on a potential project to look at getting SIL to do that. There's also LIC, which is currently used for the dairy industry, so that could be another database option that could be utilised. And then there are also private options. Currently There are a number of private different options, but currently we ourselves are developing AnyCloud, which is a genetic database that we put out there with a number of different analyses around it. But at the moment we're targeted that towards the lamb industry in China and goat milking in India. Okay, so the third component is looking at industry breeding scheme designs. So that's kind of around, I guess the key question in my mind is how fast and cost effectively can we get to where we want to go? So there are a number of factors that influence the rate of genetic progress, and they're listed on the left-hand side in genetic jargon, and the key drivers for those are listed on the right. So selection intensity is one of those factors, and one of the key drivers for that is being able to select and disseminate the best genetics. And things that I guess are drivers of that are the scale that you have to choose from. So if you've got a within flock, there will be a limited scale instead of choosing across industry-wide, and it's also dependent on what reproductive technologies we have available. So within the dairy industry, there's a massive AI program which effectively distributes a number of the best genetics through New Zealand. The second one is genetic standard deviation. So that is looking at the heritability of the trait and the variation of the trait in the population. So that's what we were talking about in the introduction. The third one is the accuracy of selection, so how good the quality of our records are, how good our parentage information is, and how accurate our recording is. Also, how good our recording has been on the relatives that have been in the past. And the last one is the generation interval, so how long it takes for the genes to disseminate throughout the population. So there are a number of different designs that can be utilised, and that's progeny testing, genome selection, or crossbreeding. Now these can be used exclusively on their own or in combination, and they're often used in combination. I'm going to just give you a brief overview of what those three are, but I'm not going to go into great detail because Suzanne Rowe is following me after this, and I think she's going to talk about the opportunities with genomic selection. And I'm not 100% sure, but I think Peter Gatley and Jake Sharon are following after that, and they may be talking about this crossbreeding, the potential benefits around that. So progeny testing is basically looking at the performance of progeny from a sire, the average performance, and then using that to predict how good the sire is. And I guess the classic example is the New Zealand dairy industry. So they have a sire proving scheme. So on the left-hand side there, they use certain bulls and put them across a selected number of cows in multiple environments. The calves are then born from those cows, and then we wait to see how those cows perform. And then based on that information, that feeds back to the breeder, and then he can see how well his sire is truly represented in terms of genetic merit. The dairy industry, as I said before, is very good at effectively disseminating the genes, and they can do that through looking at ranking both breeding worth index and then doing that throughout New Zealand. So I guess the advantages for this is that it's quite a good selection intensity side of things, but the disadvantage is that it does take a while, so the bull can be kind of 7 to 8 years before his progeny feeds information about what he is truly genetically worth. Right, genomic selection. So in my simple mind, genomic selection is looking at genetic markers associated with performance traits. So in order for us to be able to do this, it's really important that we have a training population. So that's my population on the top left there. So the population must be genotyped, but also have really strong records for performance traits. So some really good recording in there. And then what we look at is we also must have, if we wanted to use some young selection rams with that, then we would genotype those rams, and basically we're looking for markers that are associated with the phenotypic traits. So if the two animals share the same markers, they're likely to be genetically similar, and also that means that they're likely to take across the favourable trait. So it's basically very good use of information from relatives. The advantage of this is that you don't have to wait for the progeny to prove itself. You can bring a very young sires into your program in order to get genetic gain. But I guess the disadvantage is that in order to do this, you really must have a very good training population to start with, and that often the individuals that you want to use do need to be related back to that training population. Okay, so crossbreeding is another option. So what we have, I guess, crossbreeding is simply producing an animal and mating two different types of species or breeds. And I guess the best example that we have of that in New Zealand is the deer industry. So where they've taken park deer and crossed them with wild deer in order to produce the New Zealand farm deer. So the major advantage with crossbreeding is you can get a real step change in terms of the trait that you're looking for. So in the deer example, by bringing park or wapiti deer in, then we got a massive improvement in growth rates of the deer in order for them to being able to be slaughtered earlier, and also it helped with the temperament of the wild deer. But I guess the one disadvantage about crossbreeding is we do have to be careful that when we bring in an exotic genotype, I guess, that we're a little bit unsure how it would perform in our environment. So we need to be able to take that into consideration and be careful when we do that. So where I think the New Zealand sheep milking improvement program potentially is I think we're emerging industry currently. We've got genetics, I guess on the genetic side of things, we've got gene pools established over the entire country. And I guess individually there are assessments going within those gene pools to have a look at how well they're doing. And in terms of management, I think we're still refining our management systems in terms of what best suits us. So which makes us kind of an emerging industry. There are a number of challenges, I think, to become a mature industry, and it just doesn't happen overnight. But ideally, we would like to get to a point where we can look at our genetic program and understand which market that we're targeting, which there's still a lot of talk about different markets being targeted currently. Developing a breeding objective that ensures the profitability on farm for producing that product. Refining the breeding structure, and looking at the genetic evaluation. So I guess some of the challenges of where we're at with that is that we don't really have a coordinated industry body to fund any development currently. So development will fall back on the entrepreneurs in the industry as of now. It can be quite difficult to imagine what a breeding objective would look like for industry, and this is because generally at this stage of the game we've got a lot of things happening with genotypes and potential changes there, and also we're looking at a number of different production systems. Entrepreneurs can also view an industry-wide thing as potentially a threat to their own marketing strategies, and that's fair enough, and that needs to be taken into consideration. So I'm just going to leave you with a couple of questions, which I'm kind of hoping my following speakers are going to address a little. Is it the right time to have an industry genetic improvement program? And if we did have a business model, what does that look like? So you can do a cooperation, and there are advantages with that type of approach. So you have scale by all working together in terms of your genetics, and you can avoid duplication costs in terms of infrastructure requirements. Remaining as competitive models, I guess it's like anything when you're running your own business, and impact, your motivation, speed and effectiveness is a lot faster. So I know myself when with contracting, if dealing with industry bodies can often be a lot slower because it has to go through a number of processes. When we're dealing with private businesses, decisions just happen a lot faster and movements move forward. And then the last one is kind of a co-opetition. So there are opportunities out there where you can potentially look at sharing costs of infrastructure, but also allowing for market differential. So So for example, you could look at database and genetic evaluations could be a shared cost, but allowing specific indexes or something to be related to individual companies. Right, that's me.
We've got time for one question. If anyone's got a question.
That's why you end on a question.
Thanks, Joe. You've set up the afternoon well. After this session on genetics, we're going into organisational structure, so thank you for your input there. Our next speaker is Dr. Suzanne Rowe from Ag Research. She's a senior scientist there and a specialist in quantitative genetics and genomics. She has also had a lot of practical experience in the dairy industry. So welcome, Suzanne.
Thank you. Right, okay, let's get this thing going. Slides, good. Okay, Joe's told you a lot about quantitative genetics. I'm probably going to repeat a lot of what she said, so there'll be a test. So, as I've been introduced, my name's Suzanne Rowe. I'm from Inverness. I've been working in various livestock breeding schemes for a long time, and I'm really interested in dairy sheep. I'm really excited by dairy sheep. I have a dairy cattle background. Don't hold it against me. But I think there are a lot of opportunities here. So I'm going to go through a project that is dear to my heart that I would really like to see launched within the dairy sheep industry. And it takes some of the best technology that we have at our fingertips now and applies it straight into an emerging industry. And I think it's a really good application. So I hope you're with me. Okay, so what I've seen over the last day or so is that people already really know what they want from a dairy sheep. And I think it exists. That perfect dairy sheep is out there because there's a lot of variation. We know that there's a lot of variation across breed. We know that there's a lot of variation within breed. We've had people speak and they've said, okay, if we have animals in exactly the same environment, they perform very differently. And we want the stars. We want the animals that have the characteristics that we're interested in, and we want that perfect dairy sheep. And if we look at thank you. If we look at what variation exists out there, there's an amazing array of mutations across all sorts of livestock species that confer just incredible adaptation. That the FAO at the moment is going across the world and it's cataloguing all the diversity that's out there. And there are some great anecdotes. You know, it's really easy to find. These Bedouin goats, a bit like the Awassi, if you starve them, they just drop their body temperature. And they just take longer to digest their feed, and they maintain their body weight, so they can cope with long periods of starvation. There's a sheep in North Ronaldsay that basically exists on seaweed. And you know, anything else would die, but it's evolved to do that. There are lots of genes out there for things like heat tolerance, and people have found these genes, like the SLIT gene, for example, and they've introgressed them into the Senepol breed, and they're using them in places where they need heat tolerance. And LIC have done some work on that recently. And this is just to say that genomic technology isn't just for these fast-paced, highly intensive industries. This is a project that's going on at the moment in Africa for the Institute of Livestock Research. They're based in Nairobi, and they found that there's a baboon gene that infers resistance to trypanosomiasis. Now, this is a hugely important issue in Africa. And they've said, okay, let's cut to the chase, and there's all this gene editing technology around, and they've said, let's have a go. Let's see if we can put this gene into cattle and produce healthy livestock, and that's what they're doing. So there's all sorts available to us. Now, I'm not saying that we're going to jump straight into gene editing, but I'm just saying that there's technologies out there that we need to be aware of and harness. So, livestock genetics, back to where we are at the moment. And I'm going to reiterate a bit of what Joe said. Typically, 50% of efficiency and productivity gains across all livestock enterprises have come from genetics. You can go back and you can look at beef cattle, you can look at dairy cattle, you can look at sheep. So sheep in New Zealand, half the number, exactly the same amount of production in the last 30 years. Cattle, 21 litres. I don't know whether that adds up to $10 or not, Joe. You do the dollars. So we know that whatever livestock breeding system we go into, that we can get those gains in efficiency and production from breeding. And we do it using recording systems. As Joe's iterated, there are different ways of producing breeding values and creating selection indices to get what we want. And there are new ways. There's genomic selection, where if something's really hard to measure across the population and every producer can't measure it, well, we have to find another way of predicting it. And we do that by using training populations and finding markers that infer that production. And there are special ones out there. There are things... Oh, that was fast. Okay. Back. Things like markers for CVM and BLAD, which revolutionized the dairy industry. You know, fertility changed almost overnight when they found these variants and started to select them out of these high-producing bulls. There's the Scrapie genotype in sheep, which is being selected for in France at the moment. There's DGAT in dairy, there's Barula, there's Infidel. There's lots of markers that we now have a handle on that have huge effects that we can use to select with. And as Joe said, it's permanent, it's sustainable, it's cumulative, but it relies on identifying elite animals accurately. We have to find the variation, and we have to find the animals we want to breed from, and there's the rub. So breeding is a pretty easy science. You know, I mean, we all say we're quantitative geneticists and it sounds fancy. It isn't hard. There isn't much to it, basically. There's only a few factors involved. And the rate of genetic gain depends on how much the trait is controlled by genetics, the heritability. Well, most milk traits are 30 to 40%. So you pretty much know that 30, 40% of the variation that you see in your milk and composition traits is going to be, you know, controlled by genetics. We need to know how much better the candidates are from the rest of the population, and that's the data thing, that's the measuring thing, and what's been reiterated again, accurate measures. And how accurately we identify these candidates basically drives the progress, together with the generation interval, how quickly we can turn over those parents. So this is what we do when we apply genomics. And I think, have I got a pointer? Maybe that's it. Aha. Okay. So if we have an animal here that has 2 copies of a gene, and we go along these variants, and if we imagine that 2 strands of DNA are just a barcode, they're 3 billion variants long, and there's about 26 million changes that make a difference. The rest just tells us it's a sheep. And what we're trying to do is narrow down the ones that matter and use them. So if we go along here and for example, we've got a ram and it's got this barcode and it's going to give 1 copy to offspring or the other copy to offspring, generally the variants that are close together are inherited together. So if we go along here and this ram's got this absolute star variant, if you pick this one, you know you're going to add 10 litres overnight, and everyone's excited. But lurking here is this lethal recessive. Now, if we scan genomes all over, we find these lethal recessives or these, you'll pick up a variant and you'll look at it and you'll say, okay, there's a whole heap of homozygotes at one end, so they have 2 forms of this allele, and then there's a mixed one where they have 1 form of one allele and 1 form of the other, but there's none of the other form. So there's something strange. You never get 2 of these together. And when we look at these and we see this frequency issue, then we think, okay, that there's something lurking there. And basically what happens is when animals inherit 2 of these at the same time, they generally die and we don't measure them. So we're after these. We're after these all the time because these are the important ones. These are the ones that make the difference. These are the ones that affect fertility and fecundity. And very often they lurk alongside one of these, and that's when it gets important. And again, just as Joe was saying, the only way that we can do this is by using training populations and go along each variant at a time and saying, well, what's the effect of inheriting the blue form of the gene or the green form of the gene? And you know, we bob along one at a time and we do all these tests, and what we're really trying to do is cut it down and do less tests. Because the more tests we do, the less power we have to find things. So we make faster progress when we do this, when we know exactly what we're selecting for, when we can select precisely and we can identify these variants. And that's what we've done today. And it's been really successful, and it's been going really well. So we measure the DNA, we find the variants, we use them to predict, you know, and we see genetic progress because we're predicting this phenotype or this biological function. But we never seem to catch all the variants. We know that there's a heritability of 40%, and we'll go along and we'll measure the DNA and we'll add it all up and we'll say, well, where's the rest? You know, we've accounted for so much of the variation, but hey, we're missing stuff here. And in some circles, they call it the missing heritability. And the answer is, there's a whole heap in the middle that we absolutely ignore, and until now we've had to ignore it. We've had really little choice. We measure changes in DNA, but DNA isn't the whole story. Just because we've sequenced the barcode and we know what the barcode is, it doesn't mean that we now know exactly what we've got. We thought we would, and there was millions and billions spent on sequencing the human genome. Unfortunately, there's another step. So DNA is expressed, and then it's translated, and proteins are made, and these proteins are modified or folded in various ways to make metabolites, and these metabolites give us biological function. Now, you can imagine we're doing a great job at this end and this end, but missing out this in the middle has to be to our detriment. There have to be benefits in capturing this information, and that's what's happening at the moment. And the reason we haven't been able to do it so far is we haven't had decent genome assemblies, and we haven't had methods to capture all these steps. Well, we've run out of excuses, because now we've got the genomes and we've got the methods. And actually, New Zealand's very good at dairy. It's also extremely good at genomics. So if you look at genomic tools in sheep, they've nearly all been developed within New Zealand, within the International Sheep Genomes Consortium. You're very good at producing tools, and those tools are adopted very quickly all over the world. So we've got the method. We're at the start line. What next? If we know what all these things are doing, all these variants that we didn't know about, these promoters, these regulatory things, these things that are transcribing the DNA, then we can narrow down that pool that I talked about earlier. Those 26 million variants that we have, we can narrow them down, and we can do less tests, and we can home in on what's going on much, much more quickly. And we can have a better understanding of the biology behind it. Right now, we can interpret those genome scans better. Medium term, we can actually use those scans to improve the accuracy of prediction by by prioritization and by looking at this functional and biological information. I'm going to leave that. The dream is to interpret the whole genome sequence, to have the whole shebang, to know the whole picture. And so that when we go in and we have this genome, we can do a few sums on the blackboard, and hey presto, we know exactly what we're getting out of the other end. And we thought we were going to do that with DNA sequence, but we're further behind than we thought, despite the successes that we've had. So no one's going to do this alone. This is very clearly a huge project that benefits all species, because most genes, if you take their function, have that function or a very similar function across all organisms. So there's a coordinated program going on across the world where they've taken these assays and said, right, we're going to use these tissues, we're going to use these assays, and we're going to look across lots of different livestock species, and we're going to put the information together, and we're going to have a catalogue of really, really exciting and useful information. And these are the people involved at the moment, and all the major livestock industries are in there from all across the world. The only people that are missing at the moment is us. And actually, New Zealand is a big part of the International Sheep Genomes Consortium, and there are at least 3 New Zealanders who wrote this project and are on the white paper that described it. So it's not that we don't have the capability in New Zealand or the know-how. We've got it, we just haven't engaged yet. We just haven't picked up and gone with it. And you knew what was coming. I think the dairy sheep sector is an ideal opportunity to join this project, because we have health and production traits, we have yield, we have composition, we have somatic cell count, we have all these traits are really, really interesting in terms of tissue biopsies and gene expression. We can measure it in thousands of animals are already being measured. If we find the underpinning genes, we can use them immediately in our own system, and we need a means of selecting those robust high-performance stock. So I think this is a really nice fit, because we need increased production in the dairy sheep sector, but not at any cost. There are big issues. There are big issues with pneumonia. There are big issues with facial eczema, with sun tolerance. We need to know which genes infer this resistance so we can have robust, healthy sheep. We know that just increasing production by itself, we know that we can go and get high-yielding animals. But just like we were told before, sorry, this is a bit fast-paced for me, that we're not going to cope by just breeding for yield alone. We have to know what these underlying health traits are. And it's really hard to measure them. No one is going to measure them all on their own farm. No one is going to go out and find the fatty acid profile that's perfect for their sheep. Okay, we need to do that as a collaborative group, so as then we can go and say, okay, we know how to predict the fatty acid profile in our sheep because we've got a training population that tells us how to predict it. And we want to identify those lethal variants that are lurking within the dairy sheep sector. And also food safety. You know, the French are selecting against the Scrapie genotype. There'll be other zoonoses that turn up. There'll be other genes that people say, okay, we want to know the frequency in our population. And food safety is really important. And given that the consumer that you're looking at, I think that's going to be a really important factor. So I think we can add considerable value and precision to selection by incorporating this additional genomic information. But let's go back a step. We probably need around 15,000 phenotypes. Now, if we apply for money to collect 15,000 phenotypes, to do very fancy genomic analyses, we've got 15,000 phenotypes. You can forget the genomic analyses. You can put them to one side for a moment. The opportunity that offers the industry by itself is just fantastic. It's incredible. So if you just think about that sort of training population and what you can get out of it, before you move into the fancy stuff, then that for me sells it by itself. We can then go in and scale up these milk composition phenotypes that we've been looking at. We know that you've started to differentiate New Zealand milk as a different product. We also know it needs to be scaled up. We know that we've only measured it in a few animals. We need to measure it in a lot more. So if we have a project that phenotypes a lot more of these animals and adds that into this phenotype pool, then we've just got this amazing resource going. If we add in genomic information and we genotype all these animals, well, the immediate thing that every person in this setup gets out is genotypes. So they've already got a DNA profile for their animal. And DNA profiles can provide you with a pedigree, a breed estimation. Before you even think about genomic prediction, you can use them for breeding value estimation. So you have a phenotypic resource, you've got the beginnings of a training population for genomic prediction, and then we move into the assays that will move us forward into the FOUND project and the alignment with the international. And here we have 100 animals which will be looking at chromatin accessibility, and these are really fancy analyses where we really trim it down and we think very carefully about what we're doing and how many animals we use because it's extremely expensive. So to do this, we're going to need a lot of scientific capability across the quantitative genetics. We need someone that knows how to put the genotypes and the phenotypes together and do this scanning, look at these variants, look at the effects. We need molecular genetics. We obviously need bioinformatics. We need someone who's good at statistical analysis. We need to do milk protein profiling. And all this has already been done within the other MB project, so it's really easy to plug that back in here. And at Infomay, we have a really strong team that covers this. So we're happy we have the capability to take this forward. At LIC, there's a lady called Chris Caldry. She's a methylation specialist. She's very, very good at what she does. She worked at AgResearch for a number of years. We would pull her back in to do some of these methylation profiles and some of this adaptation work. We'd also be looking at immune response. We'd be working with partners such as Abacus Bio and Massey University, because if you as a producer join this scheme and you've got phenotypes and you've got DNA profiles, some of you will know exactly what to do with that. Some of you will be away. Others will be sitting there saying, well, what do I do next? And so part of this bid would be to have access to people who can help, who can tell you what you might be able to do next. And finally, we've got another little side project going on where we're really quite interested in the idea of taking some of these phenotypes here and maybe looking at sheep milk-fed infants and doing some microbial sequencing on their feces. But I've put that in as I'm going to leave that for another day. So, what do we need? Phenotypes and DNA samples, that's it. So Just the stuff you're recording at the moment and an ear punch. That's all I need from you, and a contribution of $25 per ewe. What do you get? Immediately, you get a DNA profile, which means you can work out the relationships in your flock. It means you can do parentage if you want to. It means you'll know breed composition, which is important for estimating breeding values if you're doing any crossbreeding. You'll have some known variants already, because as I've showed you, we already know things like Scrapie, we know Barula, there's a lot of variants that we've already identified. You'll be able to create your own breeding values or have access to advice to help you create breeding values. And this is just showing what we've been doing with breed composition, where we have international standards from around the world. These are ISGC samples. These are samples from Inverness, some Texel and some Romney. These are some East Frisian animals. And if you take a DNA profile and you stick it on a plot, it beautifully maps. It's really easy if you take a DNA profile to see where which breed the animal maps closest to. So these purple animals here are a flock that I was asked to identify the breed. These here are pure East Frisians. It's pretty obvious that these purple animals are also East Frisians. These green triangles are another flock, and it's pretty obvious to see that some of them are very close to East Frisian, others are closer to the Poll Dorset. So these are just simple things that we can do with the DNA profile that comes out. So that's the short-term gain. The medium-term gain will be genomic breeding values. It will be knowing those lethals, selecting them out of the population before they get to be a problem. It will be knowing those desirable variants. So if we find a DGET equivalent or a big allele that affects composition or gives us the fatty acid profile, we know that we can select for it within New Zealand. And in long term, it's being part of that state-of-the-art genomic tool development, which New Zealand has been so good at in the past and continues to be, and developing them specifically for New Zealand sheep. So at the moment, we have a low-density chip. It has 15,000 variants on it. And some of them are for parentage, some of them for alleles that have been identified. Quite a lot of those alleles have come from around the world. New Zealand's developed it, but there are French dairy sheep alleles on it. There are, meat sheep from... So I think it's time that we made sure that the next chip that comes along, the next update, has something also for New Zealand dairy sheep. And I'm pretty much going to leave it there, I think, and take questions if you have any.
Any questions? Comments? Questions? You mentioned at the very start of your presentation that we've halved the numbers of our stock, but we've doubled the production, or we've managed to sustain the same amount of meat production. I'm hoping that in a few years' time, with your research, we'll be able to double the flock size of sheep milking stock and quadruple the production. Do you think that's possible?
So, quadruple in what? How long?
Double and quadruple, so half again.
How
How long would that take?
So the French system records... Sorry. The French system are recording 65% of their ewes and 55% of their flocks in the same scheme. So this is for the lacoons. They're adding 5 litres per ewe per year. We'll probably move faster in the first few years because we're already behind and production is low. So quadrupling production is a pretty tall order, but doubling production in 10 to 15 years, I don't think would be an issue. But I'm guessing. We really need to know what we've got in there and what variation that there is. But yeah, even by simple breeding, I'm sure we can get extremely far quite quickly.
Fantastic. Thank you for your time, Suzanne. I'd now like to invite Angela Snell to the stage from MPI. Angela is the Senior Advisor for Animal Imports, and she's been at MPI for 4 years. Prior to that, she studied veterinary science in the USA, and she's got a master's degree in international political economy.
Hi, everyone. Thanks for the opportunity to come here and speak, and sorry about my late arrival. Thank you to Joe for stepping up. See if I can figure out this here. So, As you heard, I'm a senior advisor with the Animal Imports team with MPI. So my team operates pretty much under the Biosecurity Act of 1993. Specifically, thank you. What the branch that I'm with at MPI is attempting to do is keep out unwanted organisms through the regulations that we create. Goal is to allow trade, but just to keep it safe for New Zealand. So for exclusion, eradication, management of pests and unwanted organisms. Part 3 is what we work under, effective management of risks associated with the importation of risk goods. So my team is writing regulations and coming up with management strategies for trade. What we specifically write are the legal import health standards. Those are issued under the Director General, following recommendation by a Chief Technical Officer. Our Chief Technical Officer, is Matt Stone. And as some of you know about the progression of the import health standard for sheep and goat, semen and embryos, that's what I'm going to describe a little bit of today as background, and then go into where we're at with the development of the veterinary certificates. So the way the process begins is through risk analysis. That's where we identify the different hazards that exist globally when it comes to importing what we want to import. And that's what we're trying to come up with regulations to keep out. So that's in the form of an import risk assessment is conducted. It's called an IRA, and those are available online. When that was done, there were 35 preliminary hazards identified for the imports of sheep and goat semen and embryos. And that's from any country in the world. So that's a pretty steep number. If we were trying to control for 35 organisms still, that would be very expensive when it comes to testing and the quarantine and all the different measures that would be required. The next step is that document, the IRA, which, just if you're curious, looks like one of these online. Nice lengthy document. That's taken by the risk managers, so the people on my team. And we try to come up with an actual import health standard. So risk management involves carrying out the IHS development process. It's a lovely 7-page checklist, making sure that no errors are created in the process that would jeopardize New Zealand's biosecurity. As most of you know, there is a phase in that process that's public consultation, which is where we send out our draft to everybody who might use this document at some point, or everybody who might care about what's inside this document. The process that you see here, I'm not going to go into, but I have a little outline there of how long it's taken. So it's the development. In March 2011, the risk analyses were completed. There was one general covering the bulk, or every disease except for scrapie, I should say, and then an entire separate one specifically on scrapie disease. The reason for there being a separate one is because scrapie disease is really what was considered to be the major holdup in trade from scrapie-infected countries. Then in August 2012, the draft IHSs were completed. At that point, we had semen and embryos as 2 separate documents. Those went for internal MPI review when MPI was reformatting and getting a lot of legal advice on the standards. I was with MPI at that time, although I was not the original author of the draft. I took it over in 2013 and carried it on to March 2014. I got the document out for public consultation, which some of you may have reviewed and commented on. Then in 2015, I completed what's called the review of submissions, which is also available online, and we were able to issue the IHS. So what's great is this IHS provides requirements for only 17 risk organisms. We were able to eliminate a lot of them for various reasons, such as New Zealand not having the known vector of a certain disease. 17 risk organisms is certainly more manageable than 35. Okay, so I mentioned the public consultation period. MPI always tries to engage stakeholders whenever possible. The diagram here that you see in the center, I don't know if I can point with this. Is that possible? Pointer? Don't actually, oh, there it is. There we go. So risk communication and consultation at every step in the center. So throughout the process, anytime we're making any changes to what was the original document sent out for public consultation, For example, based upon a submission, during public consultation, any change that we make has to go back through the risk team for review. So we can make, and we try to make changes as much as possible in order to facilitate trade, but it always is reviewed by our risk team. We also, for example, since I've been involved in the veterinary certificate negotiation, I've been sending out updates on the progress. And that is an example just of how we try to continue to engage throughout the process as everything is being developed from the import health standard and then beyond. So moving from the import health standard into veterinary certificate negotiation is obviously taking something that was happening within New Zealand based upon New Zealand interests and taking it to another country and saying, here are our requirements, can you meet these? It's very complex for other countries. New Zealand has very rigorous biosecurity. Partly we're an isolated country, and we have a wonderful disease-free status in a lot of cases. So it is difficult for countries. They have to look at the challenge that is ahead of them in working with us to satisfy the requirements. And they have to look at that in accordance with all of their national processes and their priorities specifically. New Zealand is a small country. It's a small market. So if they're going to do a lot of work to meet our requirements, they're not necessarily going to see a huge influx of money through export. So it does make it tricky sometimes. So when we negotiate veterinary certificates, we have the political context of the exporting country, we have the economic context, cultural context, which can be significant, and then we are all operating under SPS rules. The SPS agreement, you'll see here we have the animal health, the OIE, that's based in the EU, and one reason I have there the OIE code manual is that you'll see in the import health standard many references to this code, which is put out by the OIE. We reference that as much as possible, and what that means is that we are aligning with the international recommendations for trade, which is very good for New Zealand to be seen as aligning in that way, facilitating trade. We've done that as much as possible. In some cases, we can go under because we disagree with the code requirements and we don't think the risk is as high as they do. In some cases, we go over where we perceive the risk to be greater than what is described by the code. The manual I've indicated there outlines different types of tests that are recommended. It's a very useful document for us when we're trying to determine what tests we want to see other countries performing in order for us to be sure that the product they're sending us is clean. Okay, so getting right into the meat of this veterinary certificate negotiation. So why do we have to do this? The primary reason is that during that somewhat of a restructure in the way we were writing import health standards in 2013 meant that import health standards would be written for all countries, far different from how it used to be before, where it was written for a commodity from a specific country. Now we say we want this commodity and we want it to be able to come in from anywhere in the world, so let's write the rules for any country. It's doing the bulk of the work at once, and then knowing that later on we'll negotiate with each specific country. For countries which haven't necessarily been trading with New Zealand, they have to go through what is a country approval process. Fortunately, where we're at right now with sheep genetics is we have countries which are already involved in trade with New Zealand. So that's put us ahead of the game in this process. For some other countries, it would be probably an additional year in getting confidence that we could import from a country for which we currently don't trade very much. Okay, so when does this process begin? The good thing about this IHS came out in June 22nd of June 2015. The week before, we actually alerted the EU that we were ready to begin negotiation. So on it right away. In some cases, things don't happen that quickly. And we wait, essentially, until we either have importers asking for something or we have a country that just approaches us out of the blue saying they want to negotiate a veterinary certificate. So in other words, once the IHS, the Import Health Standard, is written, at any point a country can approach us for veterinary certificate negotiation. Okay, so how does this proceed? I haven't yet described that. When the Import Health Standard comes out, it's accompanied by a guidance document, also on the website. That's less legal terminology and a way to help explain in more layman's terms what's going on in terms of the requirements. At the end of that document, we have a model veterinary certificate. That model contains what we call certifiable language. So it's all of the requirements put in a language that a country could take, just copy it and paste it into their own certificate, making it their own. So trying to make it as easy as possible for countries to develop their veterinary certificate. So when a country wants to begin, the first thing I do is give them a Word version of that model. And my hope is that they will return that to me with their logo on top, able to meet everything within it. I haven't seen it work that way yet, though. So a key part of my job is developing equivalences. So fortunately, the Import Health Standard, under the Biosecurity Act, we can do this. We can create requirements, but then if somebody can't fill those, they can present us with an alternative solution to meeting the risk that's been presented, to managing the risk. So essentially what a veterinary certificate becomes is a list of the requirements and then the equivalences to the original requirements. So another aspect of veterinary certificate negotiation is actually the negotiation. So with some countries, we're able to operate pretty easily directly with that country. Some examples might be USA and Canada. It might help that the time difference isn't as extreme. Language is the same. Just an ease of ability to negotiate certificates. We find, in general, that with the EU, going through the Ministry of Foreign Agriculture and Trade is very helpful. Right now, the person we have is based in Brussels, and she's able to meet with them. Otherwise, we'd have a 12-hour time difference in trying to communicate, and there'd also be a language barrier. So she gets help right in Brussels for any language issues. She gets to know the individual people. Our goal is always to have a negotiation with the EU as a whole. What that means is less work, one certificate for all of the countries. Unfortunately, getting a certificate with the EU, there's a much bigger prioritization list that they're dealing with. So something like this isn't necessarily at the top, and we were told it would be some time, a year or two, before it would even be looked at by the EU to come up with a general EU certificate. So we obviously went with the option that would be faster to try to meet industry's goals. And that meant working directly with the UK and France, which is where we understood there to be the greatest interest in genetics. So we have found our relationship with MFAT to be key, not just in this negotiation so far, but throughout MPI, whether it's produce or vegetables or flowers or anything coming from the EU. It's been very helpful, and also for exports. Okay. So one thing I'm frequently asked for these negotiations, how to, for example, get them to move along faster, is how can the stakeholders be involved or assist in some way? And my 2 bits of advice here is to motivate the exporter to engage their veterinary authority. As a small country, like I mentioned, there's only so much we can do to make them want to engage in this tedious process, and also to get the job done. But if you have exporters in the UK or France or anywhere saying, we have a market opportunity here, and we want you to do this for us, get this negotiation done, so that we can start exporting, then that's the competent authority, the veterinary authority's job then to engage and satisfy exporter demands. So that's really the best way. MPI has importing sheep genetics at the top of its priority list right now. And doesn't mean that I can make the countries that I'm negotiating with move any faster, but the exporters certainly can. The other bit of advice is it is helpful when I get communications regarding the equivalences that might be necessary. So For example, when importers know exactly where they want their genetics from, they tend to have good relationships with the exporters or know the disease status, for example, or what tests are going to be required. And when they communicate what they need in terms of equivalences, where they can't meet the requirements, the details that I receive, sometimes it comes in a bit faster than it might from the veterinary authority in the exporting country. So it can be helpful. Let's see, what are the challenges here? I've already described the situation that New Zealand faces in terms of being a small country and not having a tremendous amount of leverage when it comes to negotiations. The other challenge I've described is our requirements. They're tough. And of course, it's all about protecting our status, our disease freedoms. The other challenge is it's a negotiation. It's a matter of back and forth, a lot of dialogue, trying to meet people halfway without jeopardizing our biosecurity. So it's inherently going to be a tough process. And how long do they take to complete? As I mentioned, it's probably the most frequent question I receive. And I can never give anybody a solid time frame. I like to provide an estimate. My hopeful estimates tend to be wrong, I'll admit. But yeah, it's a process that's entirely really up to our partner, our negotiating partner. So the other thing I wanted to mention here is what are the risks of these veterinary certificate negotiations? So one key thing is if we were to move ahead for timelines or just based on pressure, that type of thing, we would risk coming up with an agreement, for example, with the UK or France that the EU would then want to extend to all countries. So we have to be very careful in the decisions that we make, that we would be happy with those decisions being extended to all of the EU. So that's one risk. That ties in a few different risks, I guess. It's really about not meeting timelines, coming up with a bad certificate, and then the potential risk to biosecurity. So hence why it takes a lot of effort and a lot of time. So each veterinary certificate negotiation will be different. Obviously, when you're dealing with a different country, a different commodity, there's great variability in what's going to happen during that negotiation period. And veterinary certificate negotiation. So here's just a little bit of it, some more specifics. So I mentioned the UK and France were approached by MPI's MFET representative as soon as the IHS came out, actually a week before. The first comments were received, and that was the model certificates that I mentioned. They got back to me with comments on those in September 2015 because of their summer break. So that delayed things on our mind. We were hoping that we could begin this negotiation right away in June. And then, interestingly enough, Canada, I randomly received an email from Canada with our model certificate placed in their letterhead, ready to go. From our end, it wasn't ready to go, but Canada did a lot of the work kind of behind the scenes without even communicating with us. And their certificate I actually just received this morning, a new version which has incorporated all of my comments sent to them last month or 2 months ago. And so that certificate is progressing quietly, but I would say also quite well, just a different approach, and it's pretty direct with Canada, which in some ways helps and makes it it makes it easy to get questions moved back and forth. So good progress with France, UK, and Canada, and each negotiation is at a different stage, as I mentioned. So France, we have yet to see an actual certificate come through with their letterhead on it, but the UK has provided us with one. There are just some issues still to be resolved. And then equivalences are necessary for each country. So as I mentioned, I never see a certificate come back from the model that's ready to go. It always needs consideration from the risk management aspect, but also from the risk assessment aspect. And equivalences require an additional level of approval. More time. So here's a sampling of current issues. I'm not going to go through each of these in detail. I'll just randomly pick what I can actually read here. So the Q fever one, for example, Q fever is actually, they vaccinate for it. There's a public health risk associated with it. And when we came up with the requirement, we had hoped that either donors could be vaccinated or the semen and embryos could be vaccinated. But the situation in France is that some donors would test positive because they've been vaccinated. And where a country either can't or finds it too expensive to vaccinate or to test the semen and embryos, they'd like us to recognize their national vaccination program. We don't have a measure in the IHS for that, so that requires equivalence or additional consideration. But it's risk work, and it's going to take another few weeks to hopefully come up with a new requirement that we will permit for France, but knowing that once we allow it for France, we're also going to be able to open up that option for other countries that vaccinate against Q fever. I'll end it there. If anybody has any questions.
Any questions?
Ian McDonald, Excel. Just would like to get you to comment quickly on Scrapie and the current situation the Australians are throwing at us, and also quickly help us understand the difference from what your team see to what your equivalent team in Canberra see around Scrapie.
So I mentioned that there was a risk analysis conducted specifically on Scrapie disease. The outcome of that risk analysis is that we can align with international standards, and those are outlined in the OIE manual that I described in the code. We have Stuart McDermott with MPI, who's a known international expert with prion diseases, specifically with Scrapie. And obviously, the work that he did for the Scrapie-specific import risk analysis led us to be able to go with international standards. Australia has not gone through the same process in terms of getting a risk analysis performed specifically on Scrapie, looking at the most recent research. So they are not aligning with international requirements. The outcome is that New Zealand exports to Australia, once we start importing from Scrapie-infected countries, New Zealand's status will be considered changed according to Australia, because they consider us to be using requirements which are more lenient than they would. So it's not exactly that they're going to be considering us Scrapie-infected, but it's that they don't trust our import requirements, which is something that we do with our trading partners as well. We look at what kind of import requirements they have to know, how good their biosecurity is. So the outcome is that Australia has created some interim measures for New Zealand exports of semen, embryos, and live animals, goats and sheep, to Australia. So we are looking at New Zealand exports being affected until Australia hopefully conducts the same type of detailed risk assessment that we have done. The interim measures are still being considered, discussed in terms of specifically what they mean, but it is a work in progress. But it is not interfering with our ability to move along the veterinary certificates and start importing from the EU or any other Scrapie-infected area. Did I answer your question? Okay.
Thank you very much, Angela. It's great to know where you are and who you are for all of those farms out there that are looking at importing different stock from
Thank you.
That leads nicely on to our next speakers, Peter Gatley and Jake Chardon from Southern Cross Technology. Both have wasted no time in getting involved in advancing the genetic side of our industry. Peter is GM of Maui Milk and Southern Cross, and prior to his current role, he spent 22 years as GM genetics for LIC. And Jake is a geneticist, one of the country's leading geneticists, and he's worked in the deer, dairy cattle, dairy goat space, and now the sheep dairy space. Thanks, Jen.
Thanks, Lucy. Stand here where you can hear me. Look, we've got a pretty brief time slot here, so I'm going to rip through some information. Jake's not going to bother getting up and having his say, but believe me, he's had plenty of input here. His name is on the cover here because I'd have to say after well over 20 years involved in the genetics business, so most of what I know I learned at the feet of Jake Chardon. I'll give you a little bit of background on who we are. So I used to run the genetics program that Joe described earlier in the dairy industry, breeding about 3/4 of the dairy cows in New Zealand, and also came up with the idea of starting a similar organisation breeding for venison production in the deer industry. And Jake was with me right from the outset on that. Prior to that, he was leader of one of the giant global genetics companies, Holland Genetics, now CRV, owner of Ambreed in New Zealand, and has had a number of other responsibilities, including responsibility for genetic improvement at the Dairy Goat Cooperative. Southern Cross Dairy Sheep Technology is the name that we give to the entity that we created when we kicked off as a result of a little chance meeting at this conference last year, which I'll describe to you. So on the subject of what do we need and how do we get it for New Zealand dairy sheep, I think I first asked the question, well, why do we need a better dairy sheep? And the answer is that we have a lot of fixed costs in a farming operation like this. There's a lot of money tied up in the farm and the farm infrastructure. If you're going to feed a couple of thousand sheep, well, you've still got to feed a couple of thousand sheep or more. You have some labour requirements when you get up in the morning to milk with a number of people at the shed, and then again in the evening. A lot of those costs are effectively fairly well fixed. On the other hand, your milk production per ewe might range from maybe 100 litres of lactation to more like 400, which is more typical in an industry like France, where we look for a lot of learnings actually, and I'll describe more about that. So what traits do we need? We haven't got time to go into great detail about this, but it's no coincidence that the first 7 traits listed on that slide there are what constitutes the breeding worth economic index in the dairy industry. This is the dairy industry. As I think it was Jazz said yesterday, if you think you're farming sheep, well, that's true, but we believe there's a lot more in common with the dairy industry than the traditional sheep and beef industry here. And things like temperament, udder conformation are also very important, as we heard this morning from Diane. Particularly important in the first instance, we need some milk volume. We're not getting the volume. None of us in the industry with any scale are getting the volume. We're seeing some more impressive figures coming out of the likes of Andy's and Kat's operation, or Miles and Janet, I think, get some better numbers on average. Milking relatively small numbers of sheep, but when you scale up and you move to the sort of the crossbred type sheep that we have, we're not getting very impressive volumes at all, nowhere near equivalent to leading dairy sheep nations internationally. So milk volume is probably top of our list right now, but coupled with that is this consideration around longevity, or the ability to survive in our systems, which tend to be more extensive and quite demanding on sheep, and that's where we see the vulnerability of the likes of the East Friesian. Unfortunately, these things tend to be sort of inversely correlated. If you get more milk production, you tend to trade off some longevity or hardiness or constitution, or call it what you like. The economic index is a method of taking a number of different traits and actually weighting them according to their economic importance. And the pie chart I put up there is doesn't relate to any particular belief about what the weighting should be here, but it's just an example of how you might approach this problem, as is the case in other livestock industries. So we've got some cows in the picture here, so sorry to offend you with that. It does kind of take me back. I have, obviously been so heavily involved in breeding cows for so long, but, I use this slide to make the point that when you drive through the country these days, you don't see a lot of herds that are purely black and white or purely brown. You see a lot of black cows. And the reason for that is that they're crossbred cows. They're known in the industry as the Kiwi cow. In fact, they're known around the world as the Kiwi cow. And the reason why there's so many of them, there's a reason why they are the most popular breed now in New Zealand, is purely and simply that they outperform the others. That is a fact. That's measured across data sets involving millions of cows on a number of very important traits. The genetic diversity that comes from accessing the entire gene pool. I mean, with the Kiwi cross at LIC, there's a bit of Ayrshire blood in there. There's the Holstein Friesian. There's different strains of Holstein Friesian. There's Jerseys. There's everything. And on top of that, you get some hybrid vigour, which is extremely valuable in many cases, almost invariably, a significant positive additional benefit. So we very much start in this industry from a point of view of being breed agnostic, just as we were in dairy, just as we were in deer. In deer, we sampled all the genetics we could find that offered any promise of contribution to what we required economically, and we put them through the big progeny test program, and it didn't bother us where their ancestors originated from if they were delivering the goods on farm. Extending this analogy with cows a bit further, it's vital that the genetics be integrated with the farm system. So on the left you see a typical more, say, a European, North American type system, and on the right there, more of a New Zealand system. Notice a lot of black and white cows on the left there, and more multicoloured ones on the right. In New Zealand, of course, we have seen some hybrid systems develop, very few fully intensive TMR systems, but the reason why we're able to farm the way we do on a seasonal basis with cows on grass is that we have cows that are bred for that. If you actually just bring in Holsteins straight out of Wisconsin, they're not going to do very well on grass on a seasonal system outdoors year-round. So the hybrid system, picture here on the right, shows the sort of herd home type approach that's developed in New Zealand dairying in recent years. It's kind of similar to the philosophy that we bring to dairy sheep, in the sense that we envisage a system where it's still very much predominantly grazing system, but that the animals may spend a small but significant proportion of time indoors in order to provide them protection from the extremes of the winter weather and especially the summer sun. So they can graze overnight, for example. Between the AM milking and the PM milking, they could be in the shade, having a good feed, not wasting energy on walking, and then go out and graze overnight again. If you're in a facial eczema prone area, again, an advantage there. One of the key points here is sheep milk is a lot more valuable than cow's milk. So these higher input systems have been found to create some exposure, in light of what's happened with the commodity cycle and dairy cattle. But bear in mind also that the fully intensive system, fully housed, is the norm in the goat industry, where people are still quite profitable because they're not on a commodity cycle-based system. They're in a higher milk price, and they have separated themselves out from the extreme commodity fluctuations that we see in bovine. So in sheep, we see something in between. This is exactly the same calculation formula that was put up by both Suzanne and Joe. And these are the levers we have to pull, regardless of what we're selecting for. No matter what the species is, no matter what the focus is, whether it's wool or meat or milk, this is the same formula that applies. And this is how we go about it. We need to create as much genetic variation as we can by bringing in different populations of animals. Then there's a whole raft of different technologies we can apply. We see there listed genomics, for example. We use DNA parentage testing and genomics. The requirement that Suzanne described, where the industry would benefit enormously from a large number of genotypes and phenotypes being matched. We've had discussions with Suzanne around that, and our intention is to provide a large number of those genotypes, probably more than half of what Suzanne's project requires. Progeny testing, routine process for us. Embryo transfer to multiply the valuable genes from females, because they normally only have small numbers of offspring. Of course, males with artificial insemination can generate large numbers of offspring and enable us to disseminate the highly valuable genes right across the industry. So these are all the things that are applied in other industries, and we know how to apply them here. So this genetic variation, that's the one thing that is somewhat challenging, in the sense that we just can't create that out of thin air. It exists in a population, and we're not satisfied that we have enough genetic variation across the range of traits that we need. So we're interested in sourcing some additional genetic variation. So we do have the East Friesian in New Zealand, originating from a small importation, somewhat too narrow a genetic base from an ideal perspective, still extremely valuable because without it, I don't know that we'd be milking many sheep in New Zealand. And then we have other options like the Lacone from France, which we believe has got a lot to offer in New Zealand. Various features. You see how clean they are underneath here. That's not shorn, that's just the way they are. There's a number of different advantages they have. The single teat for rapid milking isn't actually one of them, it's a ram, this one here. Yeah. So this is just a very generalised example of how we might make some trade-offs. The local breeds here, we're talking about freely available local breeds like Pole Dorset or Coopworth or whatever, in their pure form wouldn't give us enough milk, but they're good hardy animals, good longevity. The East Friesian, lots of milk, pretty fragile in an extensive system if they're not looked after well. The first cross, you get some more milk and you trade off some longevity. The Lacone, we would expect, would be a little more hardy than the East Friesian in the New Zealand environment and give us plenty of milk. That would be a positive thing. But by combining these different breeds and actually selecting, as we have already in the dairy cattle industry, the Kiwi cross type animal, we believe we can get high production with good longevity, good residual survival, good hardiness, good constitution, call it what you like. So the Lacone, without doubt, the largest and most sophisticated dairy sheep breeding program in the world. Very impressive since about the 1960s. Enormous gains made, not just on production, but a range of traits, including conformation. Jake has travelled to France 3 times in the last 18 months and established the right relationships with the benefit of a contact of his over there, who's a vet that lives in the Roquefort region where the Roquefort cheese is made. And we have arrangements with the French to access these genetics. We're now working with MPI on biosecurity issues or satisfying those biosecurity concerns so that we can bring in some of these genetics. So I have to say Angela's been particularly helpful to us in this regard. We're now looking to engage with the senior management in MPI just to make sure that they fully understand the importance of this. And so tomorrow, instead of going home to see my family, Jake and I are actually driving south and we're going to Wellington and meeting with Matt Stone and our old friend Jock Allison will be meeting us there also, and Ken Cottier, who's a vet who I've spent many years working with in LIC, a specialist in import-export business, so that we can be all clear on what is the best way forward. Love the picture of the sheep there, obviously Lacone, judging by the beret. Amazing what you can get on clip art these days. But we are making rapid progress in terms of our ability to breed milking sheep with what we have in the country already. Here we have a sheep that was a big name in the 1990s, Blue 40, pictured here with a broken down sheep farmer from the King Country back in the day. This animal here had semen frozen in the 1990s and also had embryos frozen. And those are the semen and embryos that were retained by Jock Allison from the 1990s right up until this time last year at this conference when Jake and I met Jock, and he mentioned that he had these things and it was news to us at the time. And Jake immediately committed to buy the whole lot, and we had a discussion about that afterwards. And I have to say that there was never any doubt about whether the value was there, but it was still quite a major commitment. And I also have to recognise the support that we've received from Natalie, our Shanghai-based joint venture partner who you heard from yesterday, because she's backed this program right from the outset. So we purchased these 1,700 pure East Friesian embryos. Natalie gave us some financial support. We were talking to Simon Beamish also at the conference last year, and it just demonstrates what a great networking, opportunity this organisation is. We had the embryos implanted by our old friend John Hepburn and Co from Animal Breeding Services. and within a matter of weeks, and the lambs are born in September and they're running around on our farm right now. And in fact, some of them are out working. There's a few being dispatched around the countryside, different parts of the North Island, and they're going to be busy boys over the next few weeks. Their offspring, and there could be thousands of them, can be milking next year as hoggets. Their offspring, which could be 75% dairy genetics, could be milking the following year. So you can see that we're not going to be constrained in growth by access to genetics. We'd like a broader genetic base, we'd like some new bloodlines, but what we've got will actually enable us to breed big numbers of dairy sheep. This is a chart produced by Suzanne and a team when we sent some samples to Invermay to find out what we had inherited at the WKT operation in Kuruto. And you see here the purple dots, that's pure East Frisian. We've got Texel here, we've got Romney up here. The grey dots is a spread of animals from across our population at the farm where we've been milking this year. Unfortunately, the ones with the most East Frisian blood are actually rams, and most of our ewes were found to be typically perhaps 50% or even less. So it's just fantastic to be able to actually know for sure what we've actually been milking, and then to be able to correlate that information with some production information, which is what we see here. And you see quite clearly the relationship between higher East Frisian percentage at this end, higher production, falling away like that. And it's not a line that I drew on the graph, it's a line that the computer program drew as the line of best fit. Interestingly, if you extrapolate that line out further to the point where you might have some 75% or 100% East Frisian, you can see the difference. It would make 3 times as much milk basically from a milking. This is not a surprise, it's just interesting to see a real data set that supports the contention we have that we need more dairy genes in our milking sheep. So we are in a situation now where we have a number of sheep doing pretty modest seasonal totals, and some of them showing what's possible. Even without any additional genetic improvement, we just need to multiply these genes here and we can move up to a much higher standard. So if you go back to the question that was asked before, could we double or quadruple production? Yeah, certainly. I mean, if you went to a European system, clearly you can. I mean, if you're averaging 100, you can certainly go to 400. If you're averaging 150, you could go to 600 with pure East Frisians if you farm them like Europeans do. So absolutely that's possible. It's a combination of genetics and farm systems. But one thing we're entirely confident about is that we can see big gains in the short term and even bigger gains if we can access some new blood from offshore, because they've done a lot of great work over the last 50 years, as I say, particularly in France. Would love to have the opportunity to introduce some Awasi into that equation as well. Very difficult to get importation of germplasm out of the Middle East region, but there's a handful of Awasi animals around New Zealand that we're interested perhaps to sample, and perhaps we need to be talking more to our Australian colleagues, because at least there is actually a better opportunity to move germplasm across the border there. So at Waikino Station, our new farm that Natalie and co have committed to in the Western Bays, just 15 minutes up the road from the long-established Waitui Kuruto Trust Farm, that is going to be converted over the next season or so. We'll be milking about best part of 4,000 ewes there, mostly F1 crosses from our side by our East Frisian rams over very well-bred Coopworths that we've accumulated at the farm. They'll be milking there next year. They'll be fed extremely well. In the last 2 or 3 weeks, we've sprayed off about 135 hectares, which is going to go into lucerne. There's going to be a lot of plantain. There's going to be improved pasture as well. So there'll be a big improvement in terms of nutrition. There'll be some use of barns to provide these animals with the sort of care that they need. And we're looking at importing milking parlours from France, this internal rotary design, because we believe it's much more labour efficient than anything else we've seen, and we're very excited to see them working in New Zealand. So that's where we're at, And yeah, if there's any questions, happy to have a go at answering those. And if they get too technical, Jake's always right there.
Thank you, Peter.
Why Coopworth?
Why Coopworth? Well, actually, probably is a question that Jake should answer. Jock Ellison had accumulated quite a bit of experience with the various crosses with the East Friesian back in the day, and that was his strong recommendation. I certainly wasn't going to argue with Jock Ellison, but Jake, anything to add on that?
Well, there are two reasons. First reason is that Jock thinks they're contributing the most to milk production, and secondly, facial eczema is a big freebie if you use Coopworth in the North Island.
Yeah, maybe it's a question for Jake, but I just wondered how Finn's and Paul Dorset's got on in your initial analysis.
Yeah, Paul Dorset is in the literature one of the breeds that is recommended as well. I mean, we could have chosen Paul Dorset as well. We looked at volumes of animals. We had very little time to make choices. Our OAO approval came through just before Christmas, and we had to have 4,500 ewes as soon as possible. So we went for Coopworth, and the Coopworth network of breeders was easy accessible, and we had them in no time. I agree that Paul Dorset probably is a really good alternative, and we didn't explore it. Pardon? Oh, Finnish Landrace is something you would not go for, because that is one of the lowest milk-producing breeds there is. It's high fecundity and very low milk production, so that's the one we absolutely would not choose.
Anyone else? Questions for the Maui Milk crew? Sorry, Southern Cross Technology. Thank you, Peter. Lucy's had to shoot off, so I'm doing the duties now. I think we've got one more presentation from Megan. Thank you very much, Peter. That's awesome. It's absolutely amazing. Just while Megan's getting sorted out, I mean, the internal rotary, what kind of efficiencies do you think you'll get in terms of labour use compared with the existing systems we've got now, both the scale options down south and your near neighbours?
Actually, we're another member of the team here, Garth Anderson, spent a lot of time helping us get underway with the season at the WKT farm, and is now managing the conversion for us at the Waikino farm. Just come back from France, having had a look at them directly, and hey, let's hear from Garth.
Yeah, thank you. Yeah, I was lucky enough to go over to France and visit the industry, the Lacone industry over there, which supplies milk for Roquefort cheese. With the industry being around for quite a while, a associated industry has grown up with it. A lot of technology, a lot of great ideas. We think we're quite innovative in New Zealand in the agricultural sector. They're quite innovative over there as well. So we had a good look around. I'd heard and seen plants in New Zealand. I worked at WKT. We had an external rotary there. Had a number of sort of design issues, some of which we sorted out during the year. Prior to myself going to France, we also had a look at some of the other systems available in New Zealand. And there were issues associated with what we were wanting to do at Waikino. It's quite remote, getting staff, etc. So I went over there with a pretty open mind on what we were looking at. I'd heard from Jake, who had been there, and suggested these were quite good, seen some videos he had brought back. And when we went across there, it was fascinating to see how efficient these internal rotaries were. Quite different, a lot of issues associated with just some of the requirements that we have in New Zealand compared to France, but efficiency-wise of throughput, they were doing with one person basically for most of the season, easily doing 800 to 900 animals an hour. Very simple systems. Internal rotary, you're quite close to any of the cup slipping and things like that that can occur. They had a lot of different technology for removing cups and representing cups. Yeah, it was a real eye-opener. They also had some herringbone systems that were quite interesting as well. The herringbones were swingovers. In general, I saw no low-line systems, and I'd heard also before I went that if you have a high line, you know, herringbone swingover like we have in the dairy industry, too much problem with somatic cells. No issue about that over there. Everyone was a high line, somatic cells they didn't believe was a problem. They had feeding systems that they could individually feed animals in the herringbone. Again, great technology. So I recommend to anyone who may think about this industry, if it is worth going to France to have a look. It is a real eye-opener. They were very helpful. Yeah, the other thing is the French lunches, 2 hours. €15, you get we had a 6-course meal for €15, including wine. Yeah, real nice lifestyle, but very nice people, and a real eye-opener, and hugely valuable, hugely valuable.
This is a question for Peter and Jake. I noticed in the traits that you were most selecting for, there wasn't wool. And I was just interested, given that they are sheep after all, whether there is any benefit in looking at having no wool collection or better wool collection, rather than sort of, somewhere in the middle, which sort of becomes a bit of a pain in the arse rather than anything particularly profitable for anybody.
Yeah, I'll make a comment for a start. I mean, certainly as far as wool's concerned, it's well down our list of priorities. I mean, we wouldn't ignore it entirely. I mean, just like in the dairy cattle BW, there's still a count made of the value of cull cows, but it doesn't have a huge influence on your selection decisions. And if you're having to milk during lactation, like we did this year, the whole business of shearing becomes quite an issue, really. This year we made a decision on the fly to pay a premium to have sheep milked have them shorn at the milking shed rather than walk 7 k's up the road to the shearing shed. We picked out a couple of hundred odd each day, and we had somebody shear them very carefully with full bellies, big sheep, a lot of care taken, particularly around the udder. Did a great job. Happy to pay a premium for that, and if the wool pays the shearer, then that's about all we expect. Anything to add?
Yeah, I think the quality of your milk will be a lot better if there's no wool at all around the udder and the teats. So cleanliness around, or no wool at all, is really my wish around the belly and the udder. And the Lacone will provide that. So that would be great, yeah.
Right, thank you, gentlemen. One more presentation from Megan, who's just, and then so over to you, Megan.
Cool,
Megan,
thanks.
you want to introduce yourself?
Yep. Hi, my name's Megan Shultons. Some of you may know me as a taxi driver for the last 2 days, but I'm also studying a Master's in Animal Science. My supervisors, Nicholas Lopez Villalopos and Sam Peterson, are helping me to develop the prototype model for genetic evaluation of dairy sheep in New Zealand. We are working with Kat and Andy Gunson, who commercially own and operate a dairy sheep farm in Hawke's Bay. My master's thesis is about developing a breeding objective and selection index for dairy sheep in New Zealand. This will allow the selection of superior sheep to breed from and rapidly increase the rate of genetic gain in this already evolving industry. Using random regression models, we can create lactation curves, which enable us to calculate total yields for each ewe. These will be used for genetic evaluation of the dairy sheep and enable us to choose which ewes are good for milk, fat, protein, lactose, somatic cell score, longevity, and persistency. The main objective of my thesis is to develop a prototype random regression methodology to model lactation curves of milk and dairy sheep. This will enable us to predict the milk yield production at a given time after lambing, and thus provide an understanding of the milk production pattern throughout this period. The materials and methods required for me to complete my thesis include monthly herd tests for milk volume and fat, protein, lactose, and total milk solid percentages, the 93 crossbred East Frisian ewes, and random regression models using orthogonal polynomials of third and fourth order. Although my data analysis is in its very early stages, I have generated a few lactation curves to model the population average, which is the black line, a high yielding ewe, which is the green, and a low yielding ewe, which is the red line. Here we have the predicted milk yield, predicted protein yield, predicted fat yield, and predicted lactose yield for days in milk. The important thing in here is that for each animal, I can accurately obtain the accumulated yields at different days in milk, as well as the actual lactation period. Or if they do not have a full lactation length, like the little ewe here, the little red one, then we can use this model to adjust and predict a little bit of these numbers to adjust to equal a full lactation length. In addition to this, persistency of lactation curve can be obtained. For example, U35, the red one, has low yield, a short lactation length, but has greater persistency than the high yielding ewe. And the high yielding ewe is high yielding, long lactation length, but lower persistency. All of these phenotypes derived from the lactation curves for each individual ewe will then be used for genetic evaluation to obtain breeding values and used in a selection index. Thank you.
Thank you, Megan. A question? Quick. Anyone got a... Cool. Thank you very much, Megan. Great stuff.